now let me introduce our next speaker, who is Dr. Ava Amini. She is a principal researcher at Microsoft Research. She is also a co-director of the Ex Vivo project, which is an exciting collaboration between the Broad Institute and Microsoft Research. So today she will be talking about modeling and targeting cell states in cancer. So with that, let's give Dr. Amini a round more applause.
Thank you, Wengong, and thank you to the organizers for having me here. Indeed, it's my pleasure to share about this joint research initiative we have between Microsoft and the Broad Institute, where we're synergizing machine learning and experimentation for the purpose of therapeutic discovery in the context of cancer. And really, our motivation is grounded in the fact that the bedrock of cancer precision medicine to date is based on the presence of DNA mutations, alterations present in the genome that we can then recapitulate in a subset of experimental systems to then stratify patients and screen drugs and recommend appropriate treatment courses. But the cornerstone of this is this assumption that the major features that are relevant to drug response are going to be mutationally encoded. And while indeed this pipeline for precision medicine has brought us success in the design and deployment of several targeted therapies based on these alterations, the fact remains that the majority of cancer patients don't have single actionable alterations in DNA that are amenable to such a therapeutic targeting strategy. And furthermore, even if they do, we see limited durable responses due to the arising of resistance and heterogeneity in different states and phenotypes that lead to these phenotypes. And so in the founding of this initiative, we wanted to take a more holistic sense and take a step back in considering what are the inputs that actually drive the functional phenotypes and responses of cancer cells. And while current precision medicine pipelines may be aimed at the top of this kind of pyramid here, the fact is that both intrinsic features, like the alterations in DNA, as well as influences from the local microenvironment and external signaling cues, dictate how cancer cells functionally respond and the phenotypic attributes that they exhibit. And so we thought about looking at a more dynamic and more comprehensive measurement of cell state to consider a more direct way to capture these functional attributes and ultimately be able to target them therapeutically. And so kind of our operating hypothesis is that if we expand the therapeutic search space beyond DNA-based mutations to consider now groups and programs of genes that are co-expressed and dictate these functional cell states, that we could expand our therapeutic search space for discovery. And our bedrock measurement and technology that we're using to achieve this is single-cell technologies such as single-cell RNA-seq that provide a readout of cell state and give us a vast scale and complexity of data to work with. And I hope that you can appreciate by just a back-of-the-envelope estimate of the scale of data that we can generate for just one patient sample, that this equally becomes as much of a biology problem as a computer science and data and machine learning problem. And so bringing these elements together, from precision medicine to cancer cell state to the measurement technologies that we use to drive data, is what really drove us to create this initiative that we call Project Ex Vivo, to bridge biology and machine learning towards this expanded paradigm for precision medicine in cancer. And so today I want to give you a taste of both the experimental lens and the computational lens by which we're tackling some of these problems, and hopefully show you how they can synergize to ultimately enable new therapeutic discoveries targeted at cell state. And so over the past couple of years since launching this initiative, we've been really focused on two main research thrusts. One, on the experimental side, is this question of how do we actually capture cell states faithfully in the experimental systems we develop? And once we do so, does it give us an expanded therapeutic search space? And so to go into this a little more in detail, I'd like to look at sort of a case study of sorts in the context of pancreatic cancer. Pancreatic cancer model systems, whether they be cell lines or more sophisticated 3D systems like organoids, we often look and say, okay, do they recapitulate the mutational profiles that we see across a range of patient tumors? And the short answer is yes, they do. But what's really striking and interesting is if we now look a step further at transcriptional state, despite the diversity of phenotypes that we see in transcriptional state in patients spanning these two core signatures, a classical state, which tends to be more responsive, and a basal state, which is more aggressive, these diversity of states are lost in the 2D cell line models. Namely, the classical state is basically not exhibited, even though these cell lines have the mutation profiles that are consistent with the patient tumors. And so we asked, okay, if we observe these losses of cell state, what are their actual functional consequences for drug response? And it turns out that, in fact, the responses to a chemotherapy like gemcitabine that we see in these workhorse cell line models actually are opposite to those responses that we see in patients, where classical patients exhibiting the classical signature will respond to these chemotherapies, but in 2D culture, cell lines that start off classical don't exhibit the same response. And we know from prior work in our group that we can actually attribute some of these losses to the lack of a supportive microenvironment and key microenvironmental signals that rescue these cell states. And this is indeed recapitulated in our work in actually developing organoid models where we can supply some of these microenvironmental cues in the media conditions and in the construction of these models to actually recapitulate these responses. And so hopefully this convinces you that across a range of relevant drugs in this context of pancreatic cancer, correcting these models to capture cell states appropriately can give us a corrected search space for therapeutics. But how can we actually turn this now into a scalable technique that we can apply to cell lines to improve the throughput and scale at which we perform these types of drug screens? Under a big experimental effort in our group, led by grad student Evelyn Tong, we've actually developed a scalable perturbation functional genomic technique to achieve this. And that involves the overexpression and genomic integration of a library of transcription factors that we know control some of these cell states downstream. And by doing this, we can actually select for key driver transcription factors that can preferentially drive particular cell states, for example, in the context of pancreas cancer, that we can then enrich and stabilize. And indeed, you can see this kind of illustrated in these flow plots here on the left, where hits from our transcription factor library stably push cells from these model systems into the desired state. And furthermore, we can scale this right across screening a number of TFs to now rank and nominate candidate TFs by which we can drive these state changes in a systematic manner. So this gives us a sense of an experimental arsenal to basically correct our experimental models to capture these more representative search spaces where we're now faithfully preserving cell state. On the other hand, right, to complement these experimental techniques, a big question in our group and an ultimate motivation is actually to build foundational models that can learn from these data sets and others to be able to infer some of these state-based dependencies automatically, right? To enable state-to-function mapping such that we can infer liabilities and hopefully target therapies. And if you've been sort of in this space of machine learning for bio over the past year or so, perhaps it's no secret to you that there has been a tremendous race to basically try to answer this question, to develop foundational models of cells based on training from large single-cell atlases to hopefully span, the diversity of human cell biology in a comprehensive way. we took a little different approach, which is when we thought about how do we actually build these models up from scratch and generate data accordingly, can we be first principled in thinking about what data we need and what are the capabilities that we want to achieve with these models. And a couple years back, we started by looking at, okay, what are we working with? What is the starting point? And the results we found were actually quite shocking, a bit surprising, where we exposed some of the limitations and really fundamental failure modes of some of these architectures that were being proposed. And over the past year and a half or so, many others in the field have kind of followed suit to these observations that have pointed out the critical limitations of this idea of training a virtual cell or training a cell foundation model on atlas data alone. And so, again, coming back to a fundamental first principle question, we ask, how does the data strategy around these foundation models bottleneck and impact their downstream performance? And can we use these learnings to inform our own approach to develop a foundational model for cell states and their deployment in cancer? And again, in the spirit of first principled, simple experiments that answer questions, two applied scientists, Akshaya and Maddie, and a student, Alan Den-Edel, spearheaded this study where we asked a very simple question of how does scale of data affect the performance of these models? And what they did was they looked at these atlases, these tissue-level atlases, and downsampled the data to varying fractions, trained a whole suite of models on these data in a foundational style, and tested their performance across a range of tasks. And our first observation was, again, a very striking one, that actually, with very little data, the performance of these models already reached their maximum. And further, scaling and adding additional data didn't seem to improve model performance. So we expanded this, massively expanded this, looking at a suite of different models of varying complexity, looking across a range of tasks from cell type annotation to predicting the effects of small molecule perturbations, right? The ultimate task that we want to perform in the context of drug discovery. And to kind of quantitate these results and to bring them together, we defined this metric of looking at, okay, what's the minimum fraction of data that these models need to reach the maximum level of performance within 95% of a threshold? And the startling thing was across models, no matter how simple, how complex, when we look at a task like cell type annotation, the performance saturates with just a fraction of the training data. Furthermore, this is recapitulated in the relevant, perhaps a more relevant task of predicting perturbation effects to small molecules in a completely unseen, held out evaluation setting, with just 1% of the data being what drove the max level of performance. And so it calls into question, right, what are these models actually being trained to do, and can we use this insight to inform something better? So the complementary question here is now, it's not about scale, maybe it's about diversity. Can we consider data diversity as a perspective to unlock the performance of these models? And we know, looking at the analog in the context of natural language AI, that these models are not just trained on Wikipedia, they're not just trained on code, but rather it's a composition of varying sources of text data, curated in a very principled way, according to principles in terms of the tasks we want these models to be capable of, that lends their actual downstream performance. And so taking inspiration from this, we looked at a sort of a controlled model system by which we can put some of these ideas to test. And so, inspired by that approach, we thought of the hematopoietic differentiation cascade as one setting in biology where there's a clear hierarchy of cell types and biology that's going on, and there's a clear differentiation between fully differentiated cells at the bottom and sort of their progenitors at the top. And here, again, we set up another set of systematic experiments, saying, okay, if we train models in the atlas style on some number of cells, can we assess their performance. And if we do this, how does that compare to when we capture more of the diversity across this hierarchy and across the spectrum? And the comparison here was going back to that same principle that I introduced about states in the context of cancer. Well, we can also have states in other biological contexts driven by these transcription factors that control key gene expression programs. And so it turns out that a couple years ago from work at the Broad, this same library of transcription factor overexpression was applied to embryonic stem cells to control differentiation across different terminal states in a systematic way. So we thought that this perhaps could be a source of transcriptional state heterogeneity that could perhaps boost the performance of these types of foundation models. And it turns out that when we look and we train models in these comparing ways, if we look in distribution, we see some benefit of performance from this strategy. But perhaps more strikingly is when we look at a completely out of distribution context. Now, by capturing this increased diversity from these transcription factor driven states, we can drive a greater degree of predictive performance. And so looking at how this generalizes, the answer is that in some contexts, it does apply, but in other contexts, like cancer, there are still critical states that are missing by training on this non-cancer data. And so coming back full circle to the experimental piece, what we're now doing is applying these same techniques across cancer cell lines and across therapeutic pressures, environmental pressures, to build out what we're calling these state-to-function maps, where we can leverage this systematic drive of transcriptional heterogeneity through these transcription factors and screen those against a range of therapeutically relevant compounds, environmental pressures, to now understand how cancer cell state maps to function. And the obvious goal here is to leverage these data now to build and power more informed models to learn these links in a generative and a causal fashion. Ultimately, right, our goal here is to bring these pieces together and think about, just as our learnings from these computational evaluations informed our data strategy and vice versa, how we can sort of define these closed loops in other contexts and other questions that we're exploring. So hopefully that gives a flavor of some of the questions that we have been exploring in Ex Vivo, both to credential the viability of cell state as a feature that's critical to preserve in our experimental systems, to capture relevant therapeutic search spaces, but also how the limitations of existing foundation models can inform better data strategies, better training strategies for building a new generation of AI models that will be actually capable in learning state to function in the context of cancer and beyond. So with that, I'll thank you all for your attention, and very, very gratefully thank all the amazing collaborators and my project co-leads, many of whom are in the audience in the back, and thank you all for your attention.
Thank you, Ava. All right, so let's have a couple of questions. If you have questions, please come to the front mic, and we'll also moderate on the questions that are due.
Hi, Ava. It's good to see you again. A great talk. So I was really interested in this downsampling of data, and then you saw that basically there was a sort of threshold that by which you achieved a same level of predictive accuracy with a very small subset of the data, and your hypothesis was it was really the diversity of the data that was driving the predicted accuracy, not necessarily the amount of data. I'm wondering, how does one determine that empirically? Like, do you have to sort of do that experiment where you downsample, downsample, downsample till you get to that threshold? Or is there some way to understand that before even selecting your training data? So, for example, I want to predict, if this gene results in this phenotypic change, which predicts, cancer cell state. Do I need this amount of data, this amount, do you know what I'm saying?
Yeah.
Yeah.
Yeah, it's a great question, and I think the two components go hand in hand. On the one hand, a study like ours, to your point, is very valuable because it exposes kind of where we are at in terms of the signal that we can get from a data set like a tissue level atlas. But I think, to your point, you're absolutely right that you can think prior to doing such extensive evaluations about really what are the tasks that you want to be capable in, what is the biology that you want to capture, and is that represented in your training data to begin with? And so for something like perturbation, right, if a model has never seen perturbational data before, perhaps there are states that are only induced post-perturbation that the model will need to see in order to be able to capture or perform well on those states, right? We can't expect a model to, completely shift its predictive distribution all the way to
extrapolate.
something.
Yeah,
Correct.
that far.
So they go hand in hand.
Yeah, I guess I'm thinking of it from more of a pragmatic view, which is generating all those data was a huge investment in time and money.
Yeah.
How do you go into that process saying, oh, we only have to generate these data, which costs this amount of time, and you know what I'm saying?
Yeah. Absolutely. It's a fantastic question and one that we've been thinking a lot about. So we have thought through strategies using principles of like data synergy and how data mixes are constructed to think about what perturbations or what classes of data would be most informative. And so those are some ideas that we have been thinking about and developing, but it's absolutely right that one can bring a more informed approach to this strategy. There's also a whole line of work in using kind of intermittent signal from the model to then inform the data in turn, right?
Yeah.
Where some early signal you may get about an evaluation can inform the acquisition of new data points.
And
there's
those gaps.
A long line of literature on those ideas as well.
All right, thank you.
Maybe one more question?
Quick question. Thanks so much. Kind of related. So I like how you guys are investigating all these cues that we don't think about in terms of 3D tissue, but I'm also thinking about what about cell signaling states? So cells communicate often, and I'm wondering if you guys are thinking about that in your perturbation screens and how you might encode that.
Yeah, absolutely. So the question is about cell signaling cues and experiments that we're doing to capture this. We have a lot of interest in this. In particular, one scientist in our group, Jacob, has thought a lot about the context of interaction of cancer cells and immune signaling molecules. And so this is something that we're also planning in the scope of these state-to-function screens in bridging controlled transcriptional heterogeneity with how that influences response to signaling cues that are important. Yeah.
One quick.
Quick. So first of all, very amazing talk. So just have a quick question about your foundation model to predict the single cell data. So I wonder, have you guys to think about using some kind of very traditional data, like gene regulatory network kind of data to really improve these single cell predictions?
Yeah, it's a very astute suggestion about bringing more priors like gene regulatory networks. One thing I'll clarify that the evaluations we showed, these were external models, right? That we were looking at, across the spectrum of techniques out there, what are the relative capabilities and limitations? And absolutely none of these explicitly bring in a prior about a GRN. I think there are works that try to bring this prior. My personal motto has always been, any inductive bias brings an inductive bias. So to the extent that things can be as data-driven as possible is my personal, theory. But I know there are a spectrum of different perspectives which I think can be complementary.
Thank you so much. Let's give Ava a round.